At the 1966 Cold Spring Harbor Symposium on the Genetic Code, there was a sense that we had done it all. The code was cracked. And we knew in outline how DNA exerted control of living processes through the proteins it specifies. Some of the old hands decided that it was time to move beyond the study of the gene per se. Francis Crick decided to move into neurobiology. And never one to shy away from big problems, he was particularly interested in figuring out how the human brain works. Sidney Brenner turned to developmental biology, choosing to concentrate on a simple nematode worm in the belief that precisely so simple a creature could most readily permit scientists to unravel the connections between genes and development. Today, the worm, as it is known in the trade, is indeed the source of many of our insights into how organisms are put together. The worm's contribution was recognized by the Nobel Committee in 2002, when Brenner and two long-standing worm stalwarts, John Sulston at Cambridge and Bob Horvitz at MIT, were awarded the Nobel Prize in Physiology or Medicine. Most of the early pioneers in the DNA game, however, chose to remain focused on the basic mechanisms of gene function. Why are some proteins much more abundant than others? Many genes are switched on only in specific cells, or only at particular times in the life of a cell. How is that switching achieved? A muscle cell is hugely different from a liver cell, both in its function and in its appearance under the microscope. Changes in gene expression create this cellular diversity and differentiation. In essence, muscle cells and liver cells produce different sets of proteins. The simplest way to produce different proteins is to regulate which genes are transcribed in each cell. Thus, some so-called housekeeping proteins, the ones essential for the functioning of the cell, such as those involved in the replication of DNA, are produced by all cells. Beyond that, particular genes are switched on at particular moments in particular cells to produce appropriate proteins. It's also possible to think of development, the process of growth from a single fertilized egg into a staggeringly complex adult human, as an enormous exercise in gene switching. As tissues arise through development, so whole suites of genes must be switched on and off. The first important advances in our understanding of how genes are switched on and off came from the experiments in the 1960s by François Jacob and Jacques Monod at the Institut Pasteur in Paris. Monod had started slowly in science because, poor fellow, he was talented in so many fields that he had difficulty focusing. During the 30s, he spent time at Caltech's biology department under T.H. Morgan, father of fruit fly genetics. But not even daily exposure to Morgan's no longer so boyish boys could turn Monod into a fruit fly convert. He preferred conducting Bach concerts at the university, which later offered him a job teaching undergraduate music appreciation, and in the lavish homes of local millionaires. Not until 1940 did he complete his Ph.D. at the Sorbonne in Paris, by which time he was already heavily involved in the French Resistance. In one of the few instances of biology's complicity in espionage, Monod was able to conceal vital secret papers in the hollow leg bones of a giraffe skeleton on display outside his lab. As the war progressed, so did his importance to the resistance, and with it, his vulnerability to the Nazis. By D-Day, he was playing a major role in facilitating the Allied advance and harrying the German retreat. Jacob, too, was involved in the war effort, having escaped to Britain and joined General de Gaulle's Free French Army. He served in North Africa and participated in the D-Day landings. Shortly thereafter, he was nearly killed by a bomb. 20 pieces of shrapnel removed but he retains to this day another 80. Because his arm was damaged, his injuries ended his ambition to be a surgeon. And inspired, like so many of our generation, by Schrödinger's What is Life, he drifted toward biology. His attempts to join Monod's research group were, however, repeatedly rebuffed. But after 7 or 8 tries, by Jacob's own count, Monod's boss, the microbiologist André Lwoff, caved in June 1950. Without giving me a chance to explain anew my wishes, my ignorance, my eagerness, Wolff announced, "You know, we have discovered the induction of the prophage, i.e., how to activate bacteriophage DNA that has been incorporated into the host bacterium's DNA." I said, "Oh," putting into it all the admiration I could, and thinking to myself, what the devil is a prophage? Then he asked, "Would it interest you to work on phage?" I stammered out that was exactly what I had hoped. "Good. Come along on the 1st of September." Jacob apparently went straight from the interview to a bookshop to find a dictionary that might tell him what he had just committed himself to. Despite its inauspicious beginnings, the Jacob-Monod collaboration produced science of the very highest caliber. They tackled the gene-switching problem in E. coli, the familiar intestinal bacterium, focusing on its ability to make use of lactose, a kind of sugar. In order to digest lactose, the bacterium produces an enzyme called beta-galactosidase, which breaks the nutrient into 2 subunits, simpler sugars, called galactose and glucose. When lactose is absent in the bacterial medium, the cell produces no beta-galactosidase. When, however, lactose is introduced, the cell starts to produce the enzyme. Concluding that it is the presence of lactose that induces the production of beta-galactosidase, Jacob and Monod set about discovering how that induction occurs. In a series of elegant experiments, they found evidence of a repressor molecule that, in the absence of lactose, prevents the transcription of the beta-galactosidase gene. When, however, lactose is present, it binds to the repressor, thereby keeping it from blocking the transcription. Thus, the presence of lactose enables the transcription of the gene. In fact, Jacob and Monod found that lactose metabolism is coordinately controlled. It is not simply a matter of one gene being switched on or off at a given time. Other genes participate in digesting lactose, and a single repressor system serves to regulate all of them. While E. coli is a relatively simple system in which to investigate gene switching, subsequent work on more complicated organisms, including humans, has revealed that the same basic principles apply